Hello everyone, today we are going to talk about cryopreservation. So cryopreservation basically means storage at very ultra low temperature. Usually liquid nitrogen is preferred for the storage condition, and the temperature of liquid nitrogen is minus 196 degree centigrade. So cryopreservation basically is derived from the Greek word kryos, meaning frost, which literally means preservation in the frozen state. As I told you that it's a storage of biological material at ultra low temperature, that is of liquid nitrogen, which is minus 196 degrees centigrade. Now it is a cost-effective long-term conservation of genetic resources of various plant species. The science which deals with cryopreservation is called cryobiology. So let us see the steps of cryopreservation. Now these are some of the steps. That is, the first step is your selection of material, that is selection of plant material. Then we have addition of cryoprotectant, then freezing, storage in liquid nitrogen, thawing, washing and reculturing, measurement of viability, and the regeneration of plants. Now the first step is the selection of plant material. This is one of the most important part of the cryopreservation, because if you don't select a good plant material or a proper plant material, you will realize later that there was problem with this by the time you have done a large amount of an experimental setup. Now in this case, two things are important, or two factors, which is the nature and the density of the plant material. Now any tissue can be selected for this purpose. It can be meristem, embryo, ovules, anything that you can take, or you can think of the plant material. The density should be high. This is another factor which should be kept in mind. Now addition of cryoprotectants. The term cryoprotectant is basically here. The term cryoprotectant, now you see cryo is frozen and protectant is something which protects from the frozen state. So cryoprotectants are basically chemicals which prevents the cryo destruction, now, or the damage caused due to frozen conditions. Now this can be sucrose, alcohols, glycerols, DMSO, which is your dimethyl sulfoxide. Now generally what we do is that we combine two cryoprotectants and use them together for a better and more effective results. Now next is your freezing. As you can see, freezing depends on the type of the plant species you're working with. So based on this, there are four different freezing method. One is your slow, the other is your rapid, the other one is your stepwise freezing method, and the last one is your dry freezing method. Now slow freezing, the term slow is there, so that means you're slowly keeping or frozening them at a slow cooling rate. Now rapid, the term rapid is there, that means something which used to be done very fast. So it involves plunging the vials in liquid nitrogen. The temperature decreases rapidly. The next is your stepwise freezing method. So it's basically a combination of slow and a rapid freezing method. And the last one is your dry freezing, where dehydrated cells and seeds are stored. Coming to the storage, usually the temperature ranges between minus 70 to minus 196 degrees centigrade, but as I told you earlier, liquid nitrogen is the preferred source. So what we do is that we prefer storage done at temperature minus 196 degrees centigrade in liquid nitrogen. To prevent damage, continuous supply of nitrogen is done. So you have to make sure that nitrogen, what happens in a cryo tank, what happens, it keeps on evaporating. So you have to compensate them by continuously supplying the nitrogen. Coming to the thawing, which is one of the important steps. So what you are doing in this case is you usually carried out by plunging the vials in warm water bath with vigorous swirling. So basically what you are doing is that you are bringing from minus 196 degree centigrade the plant material to a warm water bath. That means it is having some plus temperature point, so you have to do it very fast, but at the same time you have to make sure that vigorous swirling is done. Otherwise there are instances where the cryo vials have bursted out, so you have to take care while dealing with this step. As thawing occurs, the vials are transferred to another bath at 0 degree centigrade. Next is your washing and reculturing. Now if you remember, when we were adding the cryoprotectants, that is sucrose, DMSO and other cryoprotectants, now you have to remove them. So that is basically the washing. So preserved material is washed few times to remove the cryoprotectant. Then what you do is that material is then recultured in a fresh medium. Next step is measurement of viability. Now since you are keeping them for a long-term storage in minus 196 degrees centigrade, so that particular temperature or condition brings about a kind of stress to the plant material. As a result, there is a possibility of death of cells due to storage stress. Thus, viability has to be found out before going to the next step. Now, it is basically calculated, as you can see from the formula, number of cells growing by number of cells thawed into 100. So basically, when you multiply anything by 100, it correlates to the percentage configuration. Next is your plant-led regeneration. Once you get a good percentage of viability, what you do is that you take those viable seeds and culture on a non-specific growth medium. And one more thing needs to be kept in mind is the suitable environmental conditions should be maintained. So this is in general the steps of cryopreservation. So let us see the application part. So as I said, that it's a low cost and ideal method for long-term conservation of genetic species. Now, disease-free plants can be conserved and propagated. Recalcitrant seeds can be maintained for a long time. Then what you can do is that endangered species can be maintained. Then rare germplasm and other genetic manipulation can be stored using cryopreservation techniques. So these are some of the applications. You can add more to it. So with this, this was all about your cryopreservation. Thank you and have a nice time.